I'd like to thank the organizers for the invitation to talk at this meeting. And I think what you're going to hear over the next 8.5 minutes is a completely different topic, but one very relevant to understanding metabolic health. So is there a pointer? Yes. So this is just an overview. We store a fat in fat depots in different parts of the body. From the aspect of metabolic health, the abdominal subcutaneous fat, particularly when it's expanded in obesity, is particularly important for affecting the metabolic capacity, the dysfunction or function of peripheral organs. Andrea will talk about liver, and I will talk on skeletal muscle. So in the muscle and in the other tissues here, you can see what are yellow droplets, which I drew surrounded by a coat. Lipid in peripheral organs, when stored, leads to insulin resistance, inflammation, dyslipidemia. It's associated with the entire metabolic syndrome and is known when it's deposited not in adipose tissue, but in places it doesn't belong really, as ectopic fat. If there's too much ectopic fat, one gets the metabolic syndrome of atherosclerosis, fatty liver disease, and type 2 diabetes. So focusing on intramuscular adipose tissue, there's a fellow named Brett Goodpester who's the expert on this. And he's showing us a picture of a young person with a low BMI, very healthy. And you can see that there is some in the yellow and around, and in the red, you see subcutaneous fat. In the yellow, you see intramuscular fat. In good places, the muscle can use it. It doesn't accumulate where it doesn't belong inside muscle cells. In a person with obesity or in a person who's unhealthy, you can see the muscle here, but you also see adipose tissue within the muscle adjacent to it. This is an extreme example, but it illustrates the point. So why do we care about lipid droplets in peripheral tissues like muscle where it doesn't belong? A woman tends to be more insulin sensitive, and they are able to use the lipids. Insulin resistant or people with the metabolic syndrome, and more likely men, have in their muscle smaller lipid droplets. They're very good at burning them, and it correlates with insulin sensitivity. One marker, so to speak, when we're taking images of the chest, we can see some muscle, and we can look at the lipid that's there. It's really quite obvious on a CT scan or other kinds of scans. And you can look at the size of the lipid droplet, how many of them, and whether they're big. And this is well correlated with insulin resistance and skeletal muscle insulin sensitivity been measured with extremely sensitive techniques. So why is this important, and how is it regulated? Adipose tissues, which I started out with, literally talk to different organs, and they do it by secreting messages packaged in what are called extracellular vesicles, which have been studied by Dr. Shira's lab extensively. And if you look at the extracellular vesicles in the circulation, and I'll point out in the circulation, we can sample it and assess the metabolic status of the patient. Adipose tissue is the major producer of circulating extracellular vesicles. And this is just, you can look at this reference in more detail, but you can see that the extracellular, my pointer died. The extracellular vesicles in the center can affect all the organs, the brain, the pancreas, the heart, the muscle, and the liver, which you'll hear about soon. The right kind drive metabolic health. The wrong kind lead to metabolic systemic dysfunction. So we have the opportunity with taking chest CTs for screening of lung cancer. We get images which are broader than you might be interested in if you were just interested in lungs. And we can see fat and subcutaneous depots. We can see where it's distributed. Is it in the muscle that you can see? Is it in some visceral fat depots? Is it in fat around the heart? The important thing to the point that I want to make here is that myosteatosis, the fancy term for fat in muscle, particularly in excess of it, predicts type 2 diabetes over a 20-year follow-up. That's work that has been done here at Mount Sinai and led by this team. You can see the red line here is the chances of getting metabolic disease, type 2 diabetes, 20 years later after the scan. That is extraordinary opportunity to see what the risk of an individual is and the population is. So this is a very busy slide, but I want to make a very simple point from it. These are images, obviously, of the whole body. And if we look here on the lower right, you can see an individual with a high score. There's a lot of fat in the body, but this individual, in contrast to this one, which has fat all over, this individual is obviously shaped more like a female and has more fat in the lower body. This is also true of men, but the scale is slightly different. What is important about this is that we can look at a patient and it's pretty obvious where the fat is mainly distributed, what the shape of the patient is. And people who are defined by what is called a lower body fat axis in this paper, which is basically pear-shaped fat distribution, they are protected from metabolic disease. So even if they have a big belly, and a lot of subcutaneous fat, which is deleterious, if they have more fat in the lower body, it attenuates the negative effect of fat stored where it is not supposed to be. That's extraordinary. I actually study lower body fat, and we're trying to discover what it secretes that can attenuate and improve metabolic health. So basic take-home messages that I think you should always think about. Body shape. Clinicians can very easily assess body shape. Is a person shaped more like a pear or an apple? You can measure waist circumference, which is a very good thing to measure because when adjusted for BMI, a very simple measurement that's in most medical records is important. We know it's genetically determined, and we know it's a predictor of metabolic disease. Not only type 2 diabetes, but also type 2 diabetes and cardiovascular diseases. So you can say that a chest scan is an opportunistic imaging of fat distribution to organs. It's very useful as a clinical and research tool, and you can use it to interrogate what the drivers are in an individual or in a population of chronic metabolic disease. Thank you very much for your attention.